Jump right into this comprehensive one-shot explainer where we're going to break down the absolute fundamental unit of all life. If you've got exams right around the corner, or honestly, if you just want to perfectly grasp how living organisms are actually put together, you are in the exact right place. We're going to map out the entire microscopic universe that makes you, me, and literally every living thing on Earth possible.
To get our heads in the game right away, I want you to think of the cell not as some boring microscopic dot, but as a bustling, wildly complex, self-contained city that acts as the ultimate building block for everything. You know, just like you can't build a mansion without laying down individual bricks first, you definitely can't build a tissue, an organ, or an entire living body without cells. Each one is a self-contained living system, and today we're taking a VIP tour of exactly how they operate.
So here is our roadmap for a fast-paced tour through this cellular system.
One is a self-contained living system, and today we're taking a VIP tour of exactly how they operate.
So here is our roadmap for a fast-paced tour through this cellular city. First, the foundation of life. Then, cell boundaries and transport, followed by a tour of the organelles. After that, we'll tackle plant versus animal cells, prokaryotic versus eukaryotic cells, and finally, cell division and cancer. Kicking things off with Section 1, the foundation of life. How did we even figure out these tiny architectural marvels existed in the first place? Well, it started way back in 1665 when Robert Hooke looked at a tiny slice of cork and named the little compartments he saw cells. Fast forward a bit to the 1830s, and Schleiden and Schwann realized that absolutely all plants and all animals are completely made of these cells. And finally, in 1855, Virchow gave us a hugely important piece of the puzzle. New cells don't just magically appear out of thin air. They arise exclusively from pre-existing ones. Now you might be wondering, why did it take all the way until the 1600s to figure this out? It really just comes down to our eyes. Human eye resolution has a strict biological limit. From a standard reading distance of, say, 25 centimeters, we can only distinguish objects that are at least 0.1 millimeters apart. Anything closer or smaller than that just blurs into a single point. Since cells are measured in micrometers, way below this limit, we absolutely need the magnifying power of a microscope to see these micrometric cities.
Moving right along to Section 2, cell boundaries and transport.
Let's head out to the city limits. How do things actually get in and out of the cell? The whole thing is wrapped in a selectively permeable membrane, which is described by the fluid mosaic model because, well, it's flexible and it's constantly moving. It's made up of a lipid bilayer. These are special fat molecules with heads that absolutely love water facing outward and tails that are repelled by water hiding safely on the inside. Embedded in this moving layer are proteins that act kind of like security guards. They're the gatekeepers, allowing only specific substances to pass through. Now, if you happen to be a plant, fungi, or bacteria, you get a serious upgrade. An extra layer of defense called a cell wall. It's a rigid layer made of cellulose to keep you upright and protected. So a crucial point for any exam is understanding how water moves across these borders, which is a process called osmosis. And osmosis perfectly answers this classic question. If you take a piece of potato, which is entirely made of cells, is understanding how water moves across these borders, which is a process called osmosis. And osmosis perfectly answers this classic question. If you take a piece of potato, which is entirely made of cells, of course, and dunk it into a highly concentrated saltwater solution, what actually happens to it?
The answer comes down to how water always moves to balance things out. It flows from a dilute environment into a more concentrated one. If you put a cell in an isotonic solution, the balance is totally equal, and nothing changes. But here is a super high-yield memory trick for you. Hypo equals hippo. In a hypotonic solution, like pure water, water rushes into the more concentrated cell, filling it up until it swells up like a hippopotamus. On the flip side, in a hypertonic solution, like our saltwater potato example, the highly concentrated salt draws the water completely out of the potato cells, causing them to shrink and become super limp.
All right, let's dive inside with Section 3, Tour of the Organelles.
Zooming into the city's inner working districts, the organelles, which are all suspended in a jelly-like matrix we call the cytoplasm, our first stop is definitely the most famous district, the mitochondria. You've heard it a million times, and you must remember it. It's the powerhouse of the cell. It generates ATP, which is the cellular energy currency, through cellular respiration. But here is what makes it super unique and a favorite exam question. It's semi-autonomous, meaning it actually has its very own DNA and its own ribosomes. It can operate somewhat on its own.
Next, let's compare our two main manufacturing factories. The endoplasmic reticulum, or ER, comes in two distinct flavors. The rough ER literally looks rough because it's studded with little structures called ribosomes, and its main job is to build and secrete proteins. Right next door is the smooth ER. It has 0 ribosomes, so it looks perfectly smooth, and it handles the synthesis of lipids, fats, and hormones.
Okay, so once those proteins and lipids are built, they need somewhere to go, right? Think of the Golgi apparatus as the cell's post office. It's a stack of flattened sacks that modifies, sorts, and packages these materials into little vesicles so they can be shipped out. And when things get old or just break down, enter the lysosomes. This is your waste management team. It's essentially a tough little sack filled with powerful digestive enzymes, always ready to clear out dead organelles and keep the cellular city sparkling clean.
Now, found exclusively in plants, we have a really special group of organelles called plastids. And incredibly, these also have their own DNA, just like the mitochondria. They're divided into 3 main types: chloroplasts, which hold green chlorophyll to make food via photosynthesis; chromoplasts, which provide those brilliant yellows, reds, and oranges to fruits and flowers to attract pollinators; and finally, leucoplasts, which are totally colorless and act as deep storage vaults for starch, oils, and proteins, usually found down in the roots.
Which brings us to Section 4, Plant versus Animal Cells. Setting up a direct head-to-head matchup to see exactly how these blueprints differ. Plant cells are basically heavily fortified castles. They have that rigid cell wall we mentioned earlier and a massive central water reservoir called a vacuole that creates internal turgor pressure to keep the plant standing tall. Plus, they're packing those food-producing plastids. Animal cells, on the other hand, are a lot more flexible. They completely lack a rigid cell wall, their storage vacuoles are very small and temporary, and they have absolutely 0 plastids. Because, well, animals don't need to photosynthesize or attract bees.
Let's broaden our view with Section 5, Prokaryotic versus Eukaryotic cells.
Beyond just the plant and animal divide, we absolutely must distinguish between the 2 major categories of cell structure across all of life: prokaryotic and eukaryotic. Think of it this way. Prokaryotes, like bacteria, are your simple one-room cabins. They are tiny, just 1 to 10 micrometers. They completely lack a true membrane-bound nucleus, so their DNA is just sort of floating in a region called a nucleoid. And they have no membrane-bound organelles inside. Eukaryotes, which make up plants and humans, are massive, complex mansions. They're much larger, featuring clearly defined membrane-bound rooms, like a true nucleus to lock up and protect their DNA. And all those diverse organelles we just toured.
Finally, let's look at growth in Section 6, Cell Division and Cancer.
How do these cellular cities actually expand their population? Well, cells divide in 2 very different ways. Mitosis is your routine maintenance. If you scrape your knee, your cells use mitosis to split into 2 perfect, genetically identical clones to repair the damage and help you grow. Meiosis, however, is a whole different ballgame. It's exclusively for sexual reproduction. It produces 4 cells, each completely genetically unique and containing exactly half the normal number of chromosomes, creating the gametes needed to form new life.
But whether you're repairing a knee or growing a new leaf, cell division has strict rules. Normally, cells are incredibly polite neighbors. They will divide and grow, but the absolute split second they physically touch the boundary of a neighboring cell, they immediately stop dividing. This crucial protective mechanism is known as contact inhibition. It is what ensures that our organs and tissues stay exactly the size and shape they are supposed to be.
But here is a fascinating and highly tested concept recently emphasized in modern biology syllabuses. It asks a pretty terrifying question. What actually occurs when this polite stopping mechanism, this contact inhibition, just completely fails?
Well, when cells lose contact inhibition, they forget how to be polite neighbors. The brakes completely fail. They just keep replicating uncontrollably, dividing over and over and stacking right on top of each other. This wildly uncontrolled growth results in a massive clump of cells known as a tumor. And it's really important to note for your exams, this isn't just a human or animal problem. This exact kind of cancerous growth, triggered by a loss of contact inhibition, can actually affect plants as well.
We've seen how cells are these fully functioning cities, complete with their own power plants, borders, shipping centers, and reproduction facilities. But if just a single missing break, like the loss of contact inhibition, can cause an entire system to spiral into a tumor, it really makes you wonder just how delicate, intricate, and utterly miraculous the microscopic city operating inside you right now truly is, doesn't it? Thanks for joining me on this explainer. Keep feeding that curiosity, good luck on those exams, and I'll catch you in the next one.